So for today, we are going to have a new lessons in genetics, and I hope you are all ready and equipped with the new knowledge and information that we are going to have this day. Under first quarter, module number 4 is entitled When Links Matter, Finding Your Roots. And the most essential learning competency that we are going to target today is to explain the different pattern of non-Mendelian inheritance. As you notice, this is the same competency we have last week because the lesson and skills you get from week 3 discussion has something to do with the 3 topics that we are going to tackle today. And speaking of past lesson, did you still remember those things? I hope so. If not, let us refresh our mind from the previous topic because they are continuation of the things that we are going to discuss today. So last time in our lesson, I have made some presentation about the oxyribonucleic acid or the DNA, which is the blueprint of life. And I also discussed about Gregor Mendel, who was known as the father of classical genetics, and how his experiment prompted other scientists to create their own observation about how the traits and characters are being inherited from the parents. I also tackled about the 2 patterns of inheritance, which are the incomplete dominance and codominance, which are still connected in our today's lesson. I hope you can remember those things. As I was saying, there are 3 lessons that we are going to have today, and these lessons are anchored on the division made module from SDO Caloocan, and here they are. Lesson number 1, we have ABO blood type. Do you know what is your blood type? You better know it. We're also going to talk about how the sex or gender of a person is determined in lesson 2. And moving on to lesson 3, we have sex-linked traits. Alright, get ready and prepare your things because the following lessons that I mentioned need deep focus and concentration so that you will understand better and appreciate the beauty of science. So for today, these are the following goals and objectives. For number 1, determine all possible combination of genes for a specific blood type and the probability of having a male or female child. Number 2, explain how sex-linked traits are inherited. And for number 3, appreciate the importance of genes in mapping the possible traits of the offspring. These are all the objectives for our today's lesson. In our week 3 discussion, I mentioned that genetics is one branch of science that deals with heredity and variation. Genetics discusses about why certain people resemble one another, most especially in family members. Many of our characters and traits are being inherited from our parents, like our eyes, nose, ears, shape of the face, skin complexion, height, and many more. But do you know that your blood type is inherited as well? Yes, you heard it right. Our blood type is also inherited from our parents. That is why we study more about the blood types and why do we have this certain blood type. So you better get ready. Non-Mendelian genetics discusses that ABO blood type are described and explained by multiple alleles. What are multiple alleles? Multiple alleles happen when more than one allele control the inheritance of a character. Multiple means many. So in other words, in multiple alleles, there are so many alleles that can control the inheritance of a character. One example of this is the ABO blood type. As you can see, there are 3 alleles that can control the character, and those are the IA, IB, and I. IA causes the emergence of immunoglobulin A on the surface of red blood cells, while IB causes the emergence of immunoglobulin B on the surface of RBC, while I, which means that there are no formation of immunoglobulins, whether A or B. Here on the screen, you can see the table for the genotype, phenotype, and description of each blood type. But take note, even if there are 3 alleles for the blood type, which are the IA, IB, and I, only 2 are present in an individual at a time. What does it mean? Look at this table. For example, you are type A. The 2 alleles present on your blood could be IA and IA if you are homozygous type A, or IA small I if you are heterozygous type A. If you are type B, the two alleles present could be IB and IB if you are homozygous type B, and IB small I if you are heterozygous type B. Take note that blood type AB are dominant, while type O is recessive. That's why A and B are written in capital letter, and type O is written in small letter to emphasize the dominance and the recessive allele. Type AB are said to be codominant with each other. To further elaborate the blood type group, take a look at this table. Here you can see the genotype, the phenotype, and the description of allele. Did you still remember the meaning of genotype? Genotype means collection of genes which are represented by letters, capital for dominant and small letter for recessive. We have four kinds of blood type: the type A, the type B, the type AB, and the type O. And in terms of type A, we have two kinds. There are homozygous type A and heterozygous type A. What is the difference between the two? When I say homozygous, there are two alleles present in the genotype which are the same. That is why it is called homozygous. And they are called dominant since on the genotype it is observed that they are written in capital letter. Another type is the heterozygous type A. When I say hetero, the two alleles present are different. One is dominant and one is recessive. That's why capital and small letter are written here. The other kind of blood type is the type B. And like type B has also two kinds, the homozygous and the heterozygous. When I say homozygous, the two alleles present are the same, just like this, 2IB. And they are both written in capital letters signifying that they are dominant allele. For heterozygous type B, the two alleles present are different, one dominant and one recessive. The third type is the AB, which are said to be codominant with each other. If you could still remember the meaning of codominance, these are the mode of inheritance wherein the resulting allele are both expressed in the offspring, just like type AB. Type A and type B are expressed in this kind of blood type. And the last kind of blood group is the type O, which is described as homozygous recessive. Homo means they are the same, and they are written in a small letter, meaning that they are recessive. And for your information, type O is universal donor. What do you mean by that? It means that any person who has type O in their blood can donate his or her blood to anyone who are in need. So using this table, we can be able to solve the sample problems regarding genetics in terms of ABO blood group. So let's have some exercise to determine the blood type of the offspring when the type of the parents are given. Please listen attentively because I will show you how to solve the problem in blood type group. And after that, you are going to have your 4 sit work for today. So you better listen, and I suggest that you take down notes and try to answer this also. And for the problem number 1, a girl having heterozygous blood type A married to a boy who is type O. What are the blood type of the children? Determine the percentage of the type A and type O using Punnett square. For the number 2 problem, we have what blood type can be found in the offspring if a mother has homozygous type A and the father has heterozygous type B blood? Determine the percentage of each blood type using Punnett square. So, let us see how well we understand the lesson. For number 1, this is the problem. And you are going to identify first the given. And the given are IA and I for type A and double small I for type O. Why do we use this kind of genotype? Because in this problem, we have a mother who is hetero. When you say hetero, one dominant and one recessive. And the father is type O, so we are going to use two small letter, which describe the recessive allele of the type O blood. And if you want to check if the given is correct, you can go back to the table that I presented you a while ago. So here in this table, you can see that IA and small I is described as heterozygous blood type A, and double small I is described as the homozygous recessive type O. So the given that we have used are all correct. Now, let us go back to the problem. After writing down the given, we can now proceed to the Punnett square. Draw the Punnett square and write down the genotype of the parents, which are capital IA and small I for type A, and double small I for the type O, and then copy small I for the first offspring, copy IA, copy small I for the second offspring, and then I here, another I for the third, and for the last one is also double small I. After this, we can now jot down the genotype of the offspring. So we can see on the Punnett square that we have capital A and small I, and we have 1, 2. So write it down 2. The other genotype is double small I, which is also 2. And for the phenotype, the corresponding phenotype of the following genotypes are heterozygous type A and type O, and they are both 2. Okay? So we have 2 type O and 2 heterotype type A, so we can say that we have 50% type A and 50% type O. But how did we get 50%? So going back to the Punnett square, you can see here that there are 4 children. And out of 4 children, there are 2 offspring who got type A and 2 offspring who got type O. So 2 divided by the total number of offspring, which is 4, multiplied by 100 is equals to 50%. That is the way we got 50%. And for the type O, the same process. We have 2 type O divided by the total number of offspring, which are 4, multiplied by 100% is equals to 50%. And after that, also give the GR and the PR. What do you mean by that? GR is what we call genotypic ratio, and PR refers to phenotypic ratio. So our genotypic ratio is 2 is to 2. Why? Because we have 2 IA small I here, and we also have 2 II here. So 2 is to 2. Just copy. And then for phenotypic, we have 2 heterotype A, and we also have 2 type O. So all you have to do is to transfer. And then you have the ratio now for genotype and phenotype. Okay, let's have another problem about the ABO blood type. What blood type can be found in the offspring if a mother has homozygous type A and the father has heterozygous type B? Determine the percentage of each blood type. So the given are IA and IA for type A, and double and IB and small I for type B. So how did we get this? Based on the problem, the mother is homozygous. So when we say homo, it is the same, both dominant like this. And the father is heterozygous. When I say hetero, two alleles which are different, one dominant and one recessive, just like this. And if you want to be sure of your given, you can now go back to the table of ABO blood type that I presented in the previous slide. So let us see. So here in this table, you can see that AA or double capital A, this is described as homozygous type A. And the heterozygous type B is dominant IB and small I. Okay, so we can see that the given we use are correct. After checking the given through the table, we can now proceed to the Punnett square. Draw the Punnett square now and write down the genotype for the parents, which are IA and IA for the mother, who is homozygous type A, and capital IB and a small I for the father. And then copy IA, copy IB for the first offspring, IA here, small I for the second one, IA and IB for the third, and IA, small I for the last offspring. There you go. We have now the four offspring and we can now identify the genotype and the phenotype. For the genotype, we have IA and IB. How many? One, two. So write it down. And then IA and a small I. There are also two offspring who has IA and I. And write down the corresponding phenotype. It is obvious that this is codominant type AB, wherein both A and B are expressed. That is why they are called codominant. And IA, small I, is heterotype A, since we have dominant and one recessive here, and there are two. So we are done with genotype and phenotype. Determine the percentage of type AB and type A. And here in the Punnett square, we can see 50% type AB and 50% type A. How do we get this 50%? Again, looking at the Punnett square, we have 1, 2, 3, 4 children. And out of 4 children, there are 2 type AB and there are also 2 type A. So meaning 2 divided by the total number of children, which is 4, times 100 is equals to 50%. The same story with type A. 2 out of 4 children multiplied by 100 is equals to 50%. How about the ratio? The genotypic and the phenotypic ratio are both the same, 2 is to 2. How did you get this? Here, type AB is 2 and type IA small I is 2. So, 2 is to 2 is the genotypic ratio. For the phenotype, the ratio is 2 is to 2 also because we have two codominant AB and hetero A. So, 2 is to 2. Clear? As you notice, there is a chance that type AB and A will be the blood type of the children. Actually, it is a 50-50% chance. Did you understand? If it is clear, please give it a try. I will give you your seat work number one, and answer this on your notebook. And I will also give you 5 minutes to finish it. After that, you can take a photo of your work, send it to me through FB, and I will present your answer or your solution into my screen, and everybody can see your answer while you are explaining it. And whoever can explain his or her answer will be given a reward, an additional points of 20 for his or her module week number 4. Okay? So the problem or your sheet work goes like this. Father is type O and the mother is type AB. So what will be the genotype and phenotype of the offspring, and also what is the percentage and the genotypic phenotypic ratio of the offspring? Your timer starts now. Let us see now the answer. IA and IB for type AB, double small I for type O, and then the Punnett square. Write it down. So we can see here the genotype for the parents and also the genotype for the offspring. And for the genotype, we have IAI, there are two. IBI, there is also two. The phenotype is heterotype A and heterotype B. Of course, the percentage are both 50% because 2 divided by 4 children times 100 is equals to 50%. So the genotypic ratio and phenotypic ratio are both the same. Okay? So I can assume that you understand Already the first lesson for this day, and that will be the cue for me to proceed to the next topic. Okay? So for lesson number 2, we are going to study how to determine the sex or gender of a child. Is it a boy or a girl? Our body is composed of 46 chromosomes, and there are 23 pairs of chromosomes. Chromosome can be somatic or sex chromosome. What is the difference between the two? The somatic chromosome are the body chromosome, which are composed of 22 pairs, while the sex chromosome is the 23rd pair. Chromosome can be an autosome or sex. An autosome does not decide the sex of an organism, while the sex chromosome does tell the sex of an organism. Again, 22 pairs for the somatic and 1 pair for the sex chromosome. And this sex chromosome is the focus of our lesson number 2. Have you ever wondered how to know the sex or gender of a child? How are we going to know if the child will be a boy or a girl? Who will determine the sex of the child? To know the answer to this question, let us see the next slide. So lesson number 2 is entitled Sex Determination. Study this diagram. What did you notice? What are the genotype for the sex chromosome of the mother and father? How many zygote do they produce? And what are the genotypes of the zygote produced? This diagram tells us the chance of having a boy and a girl in one family. As I said in the previous slide, the 23rd pair of chromosome is the sex chromosome, which is one pair. And for the girl, the pair is X and X. And for the boy, the pair is X and Y. Again, the girl has X and X chromosome, and the boy has X and Y chromosome. What did you observe? Do they have some similarity? Can you describe the resemblance? As you can see, both boy and girl have X chromosome. And here in this diagram, we can see the chance of having a boy or a girl in one family. Take a look. The mother has 2X, while the father has X and Y. If the X chromosome of the father fertilizes the X chromosome of the mother, if we will combine it, it will result in X and X. And X and X is equals to a girl or female. On the other hand, if the Y chromosome of the father fertilizes the X chromosome of the mother, it will result in X and Y chromosome, which is equivalent to a boy or male. Another scenario is if the X chromosome of the father fertilizes the X chromosome of the mother, the resulting zygote is X and X, which is a girl or female. And if the Y chromosome of the father fertilizes the X chromosome of the mother, the zygote will be X and Y, which is equivalent to a boy or male. So take note that this is only a chance. I just gave you the idea of how are we going to get the percentage of having a boy or a girl in one family. So one zygote can be an XX or can be an XY, depending upon what kind of chromosome does the father fertilize. Okay? And to understand this further, let us continue with the next slide to know how to determine the sex of the children. Based on the diagram that I showed you a while ago, an egg cell with an X chromosome is fertilized by a sperm carrying a Y chromosome or an X chromosome. And if an egg is fertilized by a sperm carrying an X chromosome, the zygote will be a female. On the other hand, if the egg cell with an X chromosome is fertilized by this sperm carrying a Y chromosome, the resulting zygote is male. Why? Because it is X and Y. Okay? So it depends upon the kind of sperm who will fertilize the egg cell. If the sperm carry if the sperm carries X, that will be a female because the resulting zygote will be X and X. If the sperm carries the Y chromosome, that will be a male because the resulting zygote, as you can see, will be X and Y. XX is a girl and XY is a boy. So, this is a 50-50% chance. There is a 50% possibility that it could be a female and a 50% chance that it will be a male. Okay? So from this photo, it is clear that the father will determine the sex or gender of a child because the father carries the X and Y. I hope it is clear to you because this information will help us to answer this. Find out the percentage of having a boy or a girl for a child. For your second seat work, I want you to find out the percentage using Punnett square. And take note that the greater the number of children, the higher the possibility in achieving the expected ratio of 1:1. So you may now start doing your seat work number 2 on your notebook. Again, you can send the photo through FB if you are done, and I will present it on the screen and then explain it to me and to your classmate so that you can have an additional point plus the fact that you can be assured that you already learned the lesson. Take note that the genotype that you are going to use are the XX for the girl and XY for the boy. Of course, using Punnett square. Okay, you may start now. Okay, let us see the answer for your seat work. We have here a Punnett square with an X and X for the girl and X and Y for the boy. For the offspring, you can see the four offspring with two boys and two girls having X and X and X and Y. That is why our percentage for boys and girls are both 50%. Because 2 out of 4 multiplied by 100 is equals to 50%. And this information will be will be needed in the lesson number 3. I hope you got it. I hope you understand about how to determine the percentage of a boy or a girl in one family. Okay? So that will be a cue for me for me to proceed to lesson number 3, which is entitled Sex-Linked Genes. Since you are now familiar with the sex chromosome, we can now proceed to this lesson. We have two kinds of sex-linked genes. It can be X-linked genes, which are the genes located on the X chromosome. That is why it is called X-linked because these genes are located on the X chromosome. Examples are hemophilia and color blindness. Color blindness happens when you cannot see colors in a normal way. Other word for that is color deficiency. It occurs when a person cannot distinguish between certain colors, and it usually takes place between greens, red, and sometimes in blue. Hemophilia, on the other hand, is a disorder that is rare. It happens when your blood does not clot normally simply because your blood lacks the sufficient amount of blood clotting proteins or the clotting factors. A person with hemophilia bleed for a longer time after an injury. Another kind of sex-linked genes is the Y-linked genes. It is called Y-linked genes because these are the genes located on the Y chromosome. Example is the hypertrichosis pinnae oris. Hypertrichosis can be observed on human ears. It is a phenotypic trait which is described as the presence of hairs on the lower part of the distal half of the helix of the ears, just like this. And since hypertrichosis is a Y-linked gene, it can only occur in male because only male has Y chromosome. And the color blindness and hemophilia can happen to both male and female because it is X-linked. We know already that both male and female has their X on the chromosome. And to further elaborate this topic, here is a sample pedigree chart, a sample figure, wherein it shows how the disorder is being inherited from parents to offspring. Pedigree chart is a diagram where it shows the occurrence or the appearance of phenotype of a specific gene from one generation to the next one. It has been widely used to illustrate how sex-linked traits are inherited, just like this. So hemophilia is one of the X-linked disorder characterized by excessive bleeding due to lack of protein factors wherein a person bleed for a longer time after an injury. It can be inherited by the children if the parents are affected by the disorder, just like what happened to this family. Here, we can see that the mother is a carrier of the disorder while the father is a normal. Why do you say so? Here is the legend below. You can use this for you to see who is normal and who is affected. So if you have this kind of shape, we can say this normal. If you want to see the normal male, it goes like this. The hemophiliac female has this kind of legend. And if it is a carrier, this is the symbol. If it is a hemophiliac male, a darker blue will be seen. Okay? So, both of the parents are normal, but the mother is a carrier. So, if she is a carrier, she can transfer the gene to her children. So, as you can see, one child is hemophiliac male, and then this normal, this also normal, and this is a carrier. The affected male marries a normal female, and they have three children, two normal and one carrier. And then the other girl marries a carrier girl, marries a normal male. And then they have three children, all normal, but the other one is a carrier. The carrier one got married to a hemophiliac male. They got four children. So out of four children, they have four, and two out of four are affected. So using this symbol, you can be able to detect who is a normal female and male and who is the carrier, and of course, who is the affected one. Okay? And since hemophilia is an X-linked trait, both female and male can be a hemophiliac because they both have X in their chromosome. But it happens more frequently in male than in females because male has only one X in their chromosome, while the females has two. So let us try to apply this information in solving one problem in genetics about hemophilia disorder. What is the problem? Mother is hemophiliac and the father is normal. And then they have four children, two boys and two girls. The two eldest boys are affected by the disorder while the two girls are both carriers. So create a pedigree chart for this problem. So what are you going to do? You better go back to this pedigree chart, check the legend, to check what legend are you going to use for the problem. If you are sure, you can now go back to this problem and then draw or create your pedigree chart. So first we have a mother who is hemophiliac. So we have to use a darker one. While the father is normal, so we have to use the lighter blue to identify that it is normal. And then they have four children, two boys and two girls. The eldest are the boys. So the boys are affected, so we have to use darker blue to signify hemophiliac males. And then the two girls are both normal but carrier. So the legend or the symbol for carrier is just like this. Okay? So there you go. You are now finished creating your pedigree chart out of this problem. That is how you have, that is the way you can illustrate the pedigree chart. Okay? Is it clear? So if it is clear, please try it. Here is your seat work number 3. Mother is normal and the father is hemophiliac. And they have 3 children. So now we have to use 3 children only, not 4. 2 boys and 1 girl. The eldest girl is the carrier, while the 2 boys are both normal. Again, I will give you 5 minutes to create your pedigree chart, just like what I've done. And after 5 minutes, please send me the picture of anyone who wants to present their answer for you to get your 20 points additional for your module number 4. Okay, your timer starts now. Let's see. For the female or for the mother, she's normal, but the father is affected, so we have to use the darker one. They have 3 children. The eldest is a carrier, so the symbol will goes like this, while the 2 boys are both normal, so the lighter blue will be used. Okay, there you go. This is your pedigree chart already. So let us now proceed to the second type of X-linked genes, which is the color blindness. Color blindness is one type of disorder that can happen on the X chromosome because it is X-linked genes. Since X chromosome is involved here, who do you think can have this disorder? We know already that both boy and girl have X chromosomes, so it means that boy or girl can inherit the disorder from the parents. But how? There's a better way to know who among the children will inherit the disorder, and that is through Punnett square. And in doing that, we have to be familiar with the genotype and phenotype of color blindness. And here they are. This is the table for the genotype and phenotype of the color blind. Let us try to solve the problem with the help of this table. I will show you. So XX is the genotype for a normal female. And a normal female but the carrier will be having a genotype which is XXC. And if the female is colorblind, XCXC is the genotype. XY is for the normal male. And if the male is colorblind, XCY will be the genotype. So take note of this table because this will help you to solve the following problems involving color blindness. So let's start. For our problem 1, a colorblind man marries a normal female. What are the genotypes for the male? What are the genotype of the female? What is the chance that the child will be colorblind? What is the chance that the son will be colorblind? And what is the chance that the daughter will be colorblind? So there are 5 answers or 5 questions to answer. So let us try to solve that using Punnett square. First, we have to identify the given, which are X and X for a girl and X and Y, XCY rather, for the boy. Using Punnett square, write down the given X and X for the girl, XC for the boy, XCY. Copy and then copy. For the first offspring, we have XXC and then X, Y for the second, XC for the next one, XXC, and then X, Y for the next. So using the Punnett square, we can be able to answer now. What are the genotype for the male? We have 2XY, 1, 2. So when we see this, it refers to normal male. In the female, we have XXC and another XXC. What is this genotype? This pertains to normal female but carrier. Let us now proceed to the percentage of color blindness. First, we have the sun. The sun has 0%. Why? When you say son, it refers to male. And look at the Punnett square. In the Punnett square, we only have two males. How do we say so? Because male has X and Y chromosome, right? And in the Punnett square, we can see only two males here. And then out of two males, there is no one who are colorblind. So, 0 divided by the total number of sun, which is only 2, times 100 is equals to 0%. How about the daughter? The daughter pertains to the female, and the female has X and X chromosome. And in the Punnett square, we only have two daughter here, and the percentage is only 0%. Why? Because according to our answers, all females are normal. So there is no one who are colorblind. So that is 0. 0 divided by the total number of daughter, which is 2, times 100 is equals to 0%. How about the children? Children are composed of son and daughter. In short, all the offspring here. It is 0 again. Why? Because, according to our answer, all offsprings are normal. So 0 divided by the total number of children, which are 4, times 100 is equals to 0%. Okay? So that is how you are going to answer problem regarding the sex-linked genes, particularly the color-blind disorder. And if you want to be sure, let us try another problem. Okay? So the next problem will goes like this. A colorblind man marries a girl who is normal but a carrier of the gene. So what are the genotype of the male? What are the genotype of the female? What is the chance that the child will be colorblind? And what is the chance that the son will be colorblind? And the last one, what is the chance that the daughter will be colorblind? Again, reading the problem is the first step. After that, you can write down the given. XXC for the girl, XCY for the boy. XXC because the girl is normal but carrier. XCY because the boy is already affected. Using Punnett square, you can be able to answer the following problem. So XXC for the genotype of the girl and XCY for the genotype of the boy. Then copy X, copy XC, and we can have the first offspring, X, and then XY for the second, XC, and another XC for the third offspring. And for the last, we have XC and Y for the last offspring. There you go. We have now completed the Punnett square. Answers for the genotype of the male, we have XY and XCY here. Got it? So, XY refers to normal male and XCY refers to colorblind male. For the female, we have XXC and XCXC. XCXC is a colorblind female while XXC is a normal but carrier. And for that, we can have the percentage of the colorblind offspring. First, we have son. The sun is 50%. Why? Look at the Punnett square. We have 2xy here. So we can say that there are two suns. Suns meaning male. So 2 out of 2 suns, there is one male who is affected. So 1 divided by the total number of sun, which is 2, times 100 is equals to 50%. How about the daughter? When you say daughter, it is a girl or female. And in the Punnett square, we can see that there are two females here. That is 50% also. Why? Because according to our answer, there is one female who is affected by the disorder. So 1 divided by the total number of daughter, which is 2, times 100 is equals to 50%. And the last one is the children. Children are composed of son and daughter. In short, children refers to all offspring, which is 50% again. Why? Because out of four offspring, we got two offspring who are affected by color blindness. So 2 divided by the total number of children, which are 4, times 100 is equals to 50%. There you go. We are done with 2 problems regarding color blindness, and I hope you are able to understand how it goes and how are you going to solve this using Punnett square, and also how to answer the following follow-up problems. And to know how well you understand this is your last seat work. A normal male marries a colorblind female. What are the genotype of the male and female, and what is the chance of the child, the son and the daughter, to be colorblind? So you can answer these questions using the Punnett square, but take note of the genotype that you are going to use. You can check it using the table that I have given a while ago. And also take it seriously, slowly but surely, because others got confused because they do it in a faster way. So it is genetics. So you have to do it slowly but surely. So again, I will give you 5 minutes to answer this. And anyone who wants to present their answer for additional points can send the pictures of your answer to the FB so that I can present it and then explain it in front of your classmate, and of course, I will be rating you if you got it correct. Your timer starts now. So let us see the answer for the seat work. Of course, the given are XC, and XY for boy and girl. The Punnett square will goes like this: XC for the girl and XY for the boy. The offspring we got are XXC and XCY rather, XXC and another XCY for the boy. So the answers are XCY, 2XCY, which means all colorblind, and then 2XXC, which means all normal but carrier. So the percentage of the sun is 100%. The percentage is 100 because 2 out of 2 boys are colorblind. Multiply by 100, we got 100. The daughter, 0%, because there is no daughter who are colorblind. 0 out of 2 times 100 is equals to 0. And then all the children, 50%, because 2 out of 4 children got colorblind disorder. Multiply by 100 is equals to 50%. Okay. So as I can see, you really understand the lesson for today, and I hope it will help you to answer your module. And before we end, I want everybody to be reminded of the following things like the Google form, which is our quiz number for this week. I will be sending the link later. And your module answers and a performance task number 4 should be uploaded in our Google Classroom. That's all for now, and I hope you gain some knowledge and information about science today that you can apply on your real life situation. And also, thank you for all who respond, who participated, and who cooperated with the discussion. That's all for now. See you next time. Thank you for listening and goodbye.